Hi, in this video we'll talk about two aspects about stem cell technology. We'll talk about three methods for generating pluripotent stem cells, and then the reverse case, which is once you have a pluripotent stem cell, getting it to develop into a specific tissue and some of the efforts in that regard. On this slide we see three ways for making pluripotent stem cells. And again, there are a lot of words, but I want to get this all on one page. For comparison, we'll have three slides after this going over each of these methods. The first method, which we know from the first video, is the isolation of an inner cell mass from fertilized embryos. At day 5, the cells are removed and plated out on petri dishes over feeder layers and cultured to create embryonic stem cells, large numbers of them. A second method is called somatic cell nuclear transfer, sometimes abbreviated SCNT, and this is a term that has been used instead of cloning, but it is similar to the method that was used to clone, for instance, Dolly the sheep. So somatic cell nuclear transfer involves the removal of a nucleus from an egg and then the placement of another nucleus that has a diploid genome into that egg. Then the egg can be stimulated to divide just like a fertilized egg, and after 5 days you'll create again a blastocyst and an inner cell mass. The third method is the induced pluripotent stem cells, and those are created from adult cells like epithelial cells. We're going to insert 4 genes in addition to the existing genome of a skin cell. These 4 genes are primarily transcription factors. They cause the expression of lots of genes which will dedifferentiate a cell, meaning take a differentiated cell and put it back in a pluripotent or embryonic state. And then these cells are able to generate other lineages and other types of tissues. Some of the issues with the IPS technology is that by putting in these 4 genes, and we'll see what they are, they can disrupt the existing gene. It can cause mutations, and some of the genes that are put in, again, because you're trying to create immortalized, infinitely replicating cells, they have cancer-causing potential, and that could potentially limit the use of these cells. So here is the standard method: a fertilized egg, 5 days of development into a blastocyst with an inner cell mass, removal of that mass, plating out on a petri dish, and allowing it to grow. For the somatic cell nuclear transfer, we have a haploid egg. Remember, it only has 23 chromosomes, 1 copy of each. Your somatic cell has 2 copies of each chromosome, 1 copy from a mother, 1 copy from a father. The nucleus is removed. It is from the somatic cell. It is inserted into an egg so that now you have an egg with activation with an electrical pulse. You have now a full diploid set of chromosomes, like the case of normal fertilization, where you have a haploid egg and a haploid sperm, creating a diploid set of chromosomes. And now this artificially created activated cell can divide and form an inner cell mass from which stem cells can be isolated. And in the case of Dolly the sheep, you also have the ability to create a new organism. This has never been done with humans. Just recently, in the past week, this method for getting stem cells has been achieved from adult humans, where researchers were able to take 2 adults out of 39 adults. So it's a low yield process, but an adult age 35 and adult age 75 were able to produce stem cells by somatic cell nuclear transfer. One of the huge advantages of this approach is that because you're putting in a nucleus from a person's somatic cell, that this will be should be immune tolerant, that these cells will have all the same epitopes that your body has, and so it would not be likely to create an immune response. These are, in a sense, personalized stem cells. If we're going to get stem cells this way to treat disease in an adult, these eggs and sperm are different from the ones that created the organism, and so that they will have the potential of generating immune responses, just like transplanted organs when introduced into an adult body. The third way, and perhaps most exciting, is one that avoids the inner cell mass entirely and instead converts adult skin cells into a pluripotent cell type by genetic manipulation. Now, the book says 3 key genes. Pretty much every other source will talk about 4 of them. Certainly the original one was these 4 genes, and others quickly followed up with these sets of 4 genes. These are all transcription factors. They're each responsible for turning on the transcription of many genes, hundreds of genes, and this will convert a differentiated cell type into an undifferentiated pluripotent stem cell type by activating the expression patterns of lots of genes. Now, mRNA studies have shown that there are differences between iPS cells and embryonic stem cells, but the key phenotypic fact is that different adult differentiated cell types can be created. There are some problems with these. Again, first, you're inserting genes into genomes. They can cause mutations when they're inserting in. These 2 genes are proto-oncogenes, which can, if aberrantly regulated, lead to tumor formation. And so there's still plenty of work to be done. Could you, instead of genes, put in proteins? If you put in these 4 proteins, then they could convert the cell perhaps to an iPS state. But since the genes aren't there, they wouldn't have continuous expression. They would just cause the conversion, and then the expression profiles will take over of the hundreds of thousands of genes that are activated, and then maintain the pluripotent state. To maintain the pluripotent state, once you have a pluripotent cell by any of the methods, you have to add a ligand, which is called LIF, or LIF is short for leukemia inhibitory factor. This starts a signaling pathway, a JAK-STAT signaling pathway, which maintains the self-renewal and the pluripotent step. If this ligand is active, then a separate pathway is active, and that will cause differentiation of the cells. Now, it's not 100% clear whether the ligand by itself does a sufficient job, because stem cells have this unusual property that they cannot be touching a plastic or otherwise engineered surface. Embryonic stem cells are in blue. There are brown cells called mouse embryonic fibroblasts, which the stem cells sit on top, and it's unclear if they're providing some form of molecule, ligand, or contact-dependent signaling molecule that is required for the stem cells. LIF by itself in a plastic dish is not sufficient for maintaining pluripotency. So there are some questions that are still being explored in terms of how to maintain these pluripotent cells. However, if you add LIF to the media and have the mouse embryonic fibroblasts, they can be maintained indefinitely. And this shows that when it happens, when you take away LIF, when you take away the feeder cells, you will start to have differentiation into many cell types. You do not maintain the embryonic stem cell-like phenotype. And if you're trying to specifically differentiate into a tissue type, there's an intermediate step called embryoid bodies that most developmental paths go through. And this is because the cell-cell contacts of the embryonic stem cells are in unknown ways implicated in the generation of these tissues. There's some stem cells that have different environments, some that are on the inside, some that are on the outside. Those are different environments, and those lead to different outcomes. And one thing that is also depicted accurately here is that you almost always get a large mixture of cell types anytime you're trying to differentiate stem cells. It's extremely challenging and really the focus of intense study to try to get pure samples, pure cell types as you're trying to develop different tissues for different applications. We'll next explore one of the early attempts to try to get stem cells to develop into particular tissues, where embryonic stem cells were then modified into embryoid bodies, and then different growth factors were added to the embryonic stem cells, and they were going to analyze what cells resulted from the application of these different growth factors. The 8 growth factors here listed here are known to, in humans, or were thought to lead to pretty specific outcomes. Nerve growth factor, hepatic growth factor, fibroblast growth factor, epidermal growth factors. So there was hopes that you could get, if not any kind of cell type, at least a specific cell type. And what they show here in a very efficient way is once this factor is added to the embryoid body, what resulted. And you can see for these first 2 factors, nerve growth factor and hepatic growth factor, not only are you not getting a simple single cell type like neural cell type, you're getting them from all 3 germ layers, ectoderm, mesoderm, and endoderm. The second class of growth factors were at least effective at not precluding endoderm development, but these 3 factors all produced cells with mesoderm or ectoderm origin. And then finally, there was one particular growth factor that had the greatest specificity that only led to cells being generated from mesoderm. But I still think everybody was very surprised at the heterogeneity of results when added these specific factors. And from this study emerged the paradigm that there's not going to be a single switch that one needs to throw to convert cells from one type to another, but there's going to be a series of molecules needed, sometimes in combination, sometimes serially, one after another, and that it's rather a complex process to coax an embryonic stem cell into a particular tissue type. And there's been a lot of progress in that regard, and we'll hear some about that in class in terms of getting nerves and muscle cells to be generated from embryonic stem cells. And this is summarizing the results of the growth factors in the previous slide. Now, adult stem cells in particular, and the most common one, the hemopoietic stem cells, there you have a better chance of getting stem cells to differentiate into cells that you need, and it's understood in greater detail how to get selective development of some of these cell types. But the hematopoietic stem cells produce the red blood cells, and the rest of these are white blood cells, so cells that carry oxygen, and also cells of the immune system and hemostasis. The platelets are really not white blood cells, they're neither. But when we look at hematopoietic stem cell, we know most of these are transcription factors or important genes that are responsible for differentiating a hematopoietic stem cell to perhaps osteoclasts or macrophages or neutrophils. This highlights the fact again that in some ways adult stem cells are easier to use. They're committed to a certain set of cell types, they're limited that way, and that many of the sequential development of these cells into these other cell types is better understood, and we're better able to reproduce these in laboratories. So one of the outstanding questions are, if you take an embryonic stem cell, how can you get it to convert to a hematopoietic stem cell and then into these other cell types? And people have sort of abandoned that idea in general. Again, looking at the IPS as an example, can you get 4 transcription factors that will take an embryonic stem cell, again, maybe just hypothetically, directly to an osteoclast? That's where probably most of the research is going on right now. And then lastly for this section is the idea of the niche or the niche. In terms of where stem cells are, there's the idea, which has a fair bit of experimental support, that if you put stem cells in the right environments, for instance, you're trying to repair a liver, if you get stem cells in the liver environment, the niche cells that are there in the liver, can help appropriately direct stem cells towards hepatocyte type results. Now, it's pretty clear that in the end, there's going to be more needed than just dumping in stem cells into a niche and hoping that everything works out. For instance, neural stem cells do not, in general, regenerate enough of spinal cords or neurons just from being in there, that they're going to have to be encouraged and coaxed but that can be done in a variety of ways with perhaps different growth factors. But the 3D environment is certainly a key contributor. Okay, thank you for listening.